Okay, we're back. This is part two of the prep. I hope there will just be these two. And basically, where we had just finished, I had talked about the ratios, the 3-to-1 ratio, the 1-to-1 ratio of the crosses that Mendel had done. And we'll come back to these and put some genotypes on them in a moment. But this is a table, this is probably the wrong table because this is from an older version of one of the textbooks, but this information will be found in any textbook. It basically shows the 7 crosses that were done with the different traits, the seed shape, the seed color, flower color, the shape of the pods, the color of the pods, where the flowers are found, and the height of the plant. And in every case, the F1, remember, which are heterozygous because the parents were pure breeding, had just one phenotype. And so that was called the dominant phenotype or the dominant trait. But the other trait appeared, that's the wrong place to put the arrow, the other trait appeared in amongst the progeny of the F2 if you allowed the F1 to self-fertilize. And you got always approximately a 3-to-1 ratio. Notice that it's not perfect, even though there's over 10,000 counted here. There's 850 or so counted here. The closest one is like 8,023. P's counted. So the ratios are not perfect, but they're all really close to 3-to-1. And so that ratio was observed consistently in the F2 progeny for the crosses. And those are the key points. So dominant trait, but the recessive one is still there. It never blended or got destroyed. It was actually present, but just not visible, but we can see it again then in the F2 generation. Okay, which is what I just said. And the other thing that is important is that he did the crosses in both possible directions. That means, the purple flowering plant was the male plant and the white flowering plant was the female one, or vice versa. And it never made any difference which was the female, which trait came from the female or the male. So these were all autosomal traits, which we'll talk about probably in the next lecture. So how can we explain these? Well, first of all, a reminder, the different forms of the same gene are called alleles, and we usually use a capital letter or sometimes a plus superscript. So we could have written the dominant allele this way or commonly this way. And in this case, it's the wild type functional allele. So in heterozygous individuals, the F1, they had one copy of the dominant allele and one copy of the recessive allele. Okay, and when a trait is dominant, it doesn't matter if you're homozygous or heterozygous, as long as you have at least one of the alleles, you're going to have the phenotype, which in this case is yellow. So we do have a way of expressing that, because when you look at a yellow pea seed, you have no idea what the genotype is. We write it like this, with the dominant yellow, because we know there's one, but this dash means that it could be a dominant or a recessive allele there, and that we can't really tell. That looks like it says yay. It doesn't. This is supposed to say or. The lowercase letter is the recessive form. In the case of these traits, all the traits, they are loss of function mutant alleles. And so the trait is only observable when both of the alleles are the recessive one, and the genotype would be written like this. In some textbooks, they don't actually put the slashes in. But it's common for us to do that in genetics, and I will be doing it all the time. So these symbols represent the different alleles of that gene. And as I said, the wild type could have also been expressed this way. So a heterozygous individual, we would write it like this, and with the slash. The slash just indicates different homologs, and I'll talk about that notation when I'm talking about two gene traits later on. Probably not until the next week. Okay, so heterozygous, you have two different types of alleles of your gene. Homozygous, both your alleles are the same, either two dominant alleles or two recessive alleles. Okay, and then this is the notation I was telling you about, and just think about why we use that. Why, in some situations, we have to give the genotype this way, and why? Why is that? I might ask that in class. Okay, so if we go back to this diagram, where do these traits come from? The yellow F1 were heterozygous, so they had one dominant and one recessive allele. If you crossed them, self-pollinated them, then we had progeny that were either homozygous or heterozygous for the yellow allele, and individuals that were homozygous recessive, and this gave us a 3-to-1 ratio. And we'll do a Punnett square shortly to demonstrate that that is the correct ratio and exactly how that comes about. Now, one thing he did that was clever and actually a good idea is he did what is called a test cross. He crossed individuals that were F1 yellow, and so he had already decided that they must have one of each of these factors, these particles, whatever they were. And so he decided to test that by crossing to individuals that were green. So they would have two of these particles or factors, but they would both be the same. And he predicted, if this is the case, we would expect that half of the time we had heterozygous individuals, and they would be yellow, and the other half of the time we would have homozygous individuals, and they would be green, and that is indeed what he got. So this was a way of testing the actual genotype up here of these guys. And so when in any kind of scientific work, it's very important that you draw a conclusion, yes, this makes sense, but then figure out a way of testing it in a different way, make a prediction, and know what should happen if your hypothesis is correct, and then make sure the results that you obtain actually fit that. That is good evidence, and that's how we roll in science. Okay, so the first law is equal segregation. So the hereditary factors, which we now know are called genes, they exist in pairs, and there's one pair for each character. Now, some traits are more complex than that, and we'll see examples of that, after the midterm. P's are diploid, and so this makes sense. So the members of those of each gene pair separate from one another during gamete formation. We now know that the members of those gene pairs are called alleles. That's the modern way of describing it. And we talk about segregation rather than separation during gamete formation. Just remind yourself, I'm trying to key you back into previous lectures by reminding you of things we've talked about before, so you can see that this is all one long narrative, one long story. So what type of cell division? So a single gamete is going to carry only one copy of each gene. So if an individual is heterozygous, half the gametes will carry one of the alleles and the other half will carry the other allele. And this seems obvious to us, but at the time, this was pretty revolutionary information. And then once you have, gametes that are, carry one or the other allele, the fusion is random with respect to the alleles. So a sperm that is carrying a dominant allele doesn't stop and check what genotype the egg is before it fertilizes it, right? The egg doesn't go, wait, you have a dominant allele, I only want recessive alleles, or anything like that. So whatever sperm and egg meet each other, that's the fusion that takes place, the connection. And so every combination is equally likely as any other combination, right? And so we'll do a Punnett square to show that in a minute. Okay, so the first law, so the alleles separating from each other is not really relevant. That law sort of doesn't matter in a way if we're homozygous for the dominant or for the recessive alleles, because if both of your alleles are the same, then all the gametes, sperm or eggs or pollen or ova, are all going to be either the recessive or the dominant trait. It's when we're heterozygous that this law is really important. And what it means is that there's a 50% chance of any particular egg or sperm having the dominant allele if we're heterozygous, and a 50% chance of it having the recessive allele. And you might want to think of this as sort of like a coin toss. I want you to think about how this might be similar. This is a special issue nickel. My dad collected coins, and he would have loved this. I don't think he ever saw it, but he would have loved it. Okay, so make this connection and think about how the being heterozygous and then how gamete formation in that heterozygote is related to the tossing of a coin. Okay, and then this just again just remind you about gametes, pollen, and ova in plants, so sperm and eggs, or ova, sometimes in animals. And only one randomly chosen factor for each gene pair is put into a given gamete. Those gametes fuse to form an embryo, and the embryos end up with 2 copies of each gene, one from each gamete, and therefore one from each parent. So I'm kind of repeating this because I'm trying to make it super clear. So it might feel boring and repetitive, but I'm okay with that if it means you understand really well. Okay, so this is how we use a Punnett square to actually predict the outcome of a cross. So here we have pure breeding yellow parent and a pure breeding green parent, both homozygous, of course. The F1 are heterozygous, and they're going to show the dominant phenotype, which is the yellow pea color. And then if we allow that to self-fertilize, it's like making a cross between 2 heterozygous individuals, right? So the way we do the F2, the Punnett square for the F2 is you take suppose this is the female parent and that's the male parent, or whatever. So we show the gametes for the female parent on one side and for the male parent along the top, or the other way around. It doesn't matter. So long as you put one parent here and one parent there. So this individual is going to have what gametes? 50% of the time, think of a coin toss again, the egg, the ova, is going to have the dominant allele, and 50% of the time it's going to have the recessive allele. So we have to start with the gametes, right? The same with the male, the pollen, half of the time it's going to have the dominant allele, and the other half of the time it's going to carry that recessive allele. Okay, those are equally likely outcomes. We couldn't calculate any of this if that wasn't the case. Okay, and so then how do we do this? We say, okay, so this ovum with the dominant Y is equally likely to be fertilized by a pollen with a dominant or recessive allele. So the outcomes could be homozygous for the dominant allele, or they could be heterozygous. And the likelihood of that is just the product of, you multiply those probabilities times each other, 1/4 and 1/4. Okay, so that's the first half. The second half is this recessive allele can equally combine with the dominant, so we could have heterozygous here, and the frequency is a half, right? Times a half. Or the 2 recessives could combine. And that's a half times a half, and then the recessive. So when we look at this, what we're going to see is this is not a very good highlighting job, but 3 of the 4 possible outcomes is being heterozygous, and or homozygous dominant, and therefore having the yellow phenotype. And only a quarter of the time are you going to have the green phenotype. And so this gives rise to, whoops, not that. I want to go back to the regular pen. And so this is going to give rise to the 3/4. And now this is where we put this notation, because when we actually look at these P's, we're not going to be able to tell the difference between homozygous or heterozygous ones. They all just look yellow. And then 1/4, that's going to be green. And this is a phenotypic ratio. But if we were looking at the genotypic ratio, it would be 1/4 homozygous dominant, 1/2 heterozygous, and 1/4 that are homozygous for the recessive trait. And I'm going to stop there because it's been 16 minutes and I don't want to rush through the next section. So we'll do one more, but the third one will be shorter than this one.